So in the talk today, I will start by listing the design requirement for LVP and delineate the ENL strategy, specifically speaking to the required data, the supporting data that will help us in defining the design and qualifying an LVP, particularly talking to the ENL study design and naming some challenges. And I will take the opportunity to present a case study specifically for nitrosamines for ENL and finalize the talk by take-home messages. So the LVP started to evolve quickly in the past few years, and this is particularly due to the increasing patient needs. Mainly that there are diverse groups of patients, and each pool of those patients are increasing, like neonates, like patients with chronic disease, patients with cancer, and now patients who are treated for COVID as well. And this is clearly reflected on the market share the LVP is acquiring in the past years and what is expected from that in the coming years. And to meet this increasing need, the LVP itself should evolve in such a way that it can house various drug products, become more sophisticated from a packaging standpoint, diverse volumes from few milliliters up to several liters, that can treat a population from different age groups, that have different dosage regimen, and can be treated for different periods of time from a few days up to a lifetime. These requirements are reflected on the design requirement for LVP, and the LVP should consider several parameters that are listed here, but not limited to, even sealing properties, appealability, sterilization, specifically that most of LVPs are sterilized by steam sterilization. It is compatible with the chemistry of the drug. The bag should be flexible. It has a good oxygen barrier, light barrier perhaps. It is compliant with pharmacopoeia, show low migration, and transparent. To be able to have all these requirements in place, a monolayer film, or the evolution starting from a glass bottle to a monolayer film, doesn't work anymore, and the need for a multi-layer film is strong, and this multi-layer film can be formed from the contact layer with a solution to the outer layer with several tie layers, maybe a barrier. This sophisticated film will definitely present a challenge on the migration of extractables or leachables into the solution. And we have to consider that in our assessment. Not only the primary packaging, because the whole system can also contain print labels, closures, maybe rubber in them, other accessories such as a secondary packaging and oxygen absorber, that we have to consider the whole system as a whole in the ENL strategy. And now the ENL strategy must cover the product life cycle from early stage in exploratory phase all the way up to launch and sustaining the product on the market, or even for the geo expansion practices. And the product should be compliant with the regulation and compliant from a material standpoint. So the ENL strategy can be implemented at any stage. However, it is highly recommended that the ENL strategy is implemented at a very early stage of the product. And we have different steps, a process step by step, how to design an LVP and how to qualify it from an ENL standpoint. And in the coming slides, I will walk you through these steps one by one. So first and foremost, the material selection. It's clear that if we select the material upfront, we know what we have in the packaging system. We will not be too surprised when the ENL profile comes in when we do the test on the final product. So it's a very critical step, and it will change a lot the ENL strategy and will position it for success. And here's a list of don'ts and do's. So Don't miss the opportunity to get as an E&L SME involved in the project at an early stage. Don't underestimate the importance of the supplier's data, and don't avoid investing in preliminary testing and wait only for the testing to be conducted at a later stage. What we can do certainly as E&L SMEs is to engage with the supplier to collect relevant info related on the design and on the application of this LVP, and have a thorough screening of the required material to feed the risk assessment. And by this, we can move to the second step of this E&L risk assessment, that we have to consider the following things. What's the packaging material of construction for each and every component of the packaging system? What is the packaging format, meaning that what is the different formats, what is the surface area and volume that we have, what is the drug chemistry, what are the manufacturing conditions, do we have filtration, what's the sterilization conditions, how long the product can stay in shelf life, what are the conditions for clinical use, and the patients that are treated, what their dosing regimen, meaning that the maximum daily dose and the period of treatment, and finally, the country of release. Why it's important to know the country of the release? Because we have to make sure that the qualification of the LVP is fit for the country. So that if the product is released in the US or in Europe, we have to follow suit, and this will help us categorize where the supplier data and the supporting data can be classified as critical, important, and supportive. So it depends on the country and particularities of the LVP system. The data in the important bucket can shift into critical or supportive. So here is just a mere example for some supporting information that we can get from the supplier on the general material. What is critical is the material physical chemical properties, of course, and safety data sheet. It will be very important to have certificate of analysis, if any, and info about manufacturing, particularly, for example, if there's adhesive added, if there's a specific reactor used, if there's a special vulcanization process followed for the rubber, and so on and so forth. From a supportive data also can be if the supplier already generated extractable data on the material, if they have on the organic sides of it, and comparability results. And in this case, it means that the medicinal companies already have the raw material from the supplier, and the supplier decided to apply a change. And if the supplier therefore provide a comparability testing results between the existing material and the new material, that will help greatly in the risk assessment of the new material. Second scope is the supplier info that can feed into the safety data, meaning what are the material of concern in this packaging system. So of course, we will have to consider the REACH document, the EU directive for TSE, PSE, certificates of absence of bisphenol, phthalates, nitrosamines, natural latex, MBT in case of a rubber, and biocompatibility data or USP classification per 88. It's also important to have substance of very high concern data sheet, the CMR compliance for EC 1272, and other perhaps biocompatibility data concerning USP 87. Sometimes also it can be supportive to have the California Proposition 65 or the PAS. And if we talk about regulatory aspect, the data that we need to have the regulatory compliance, and this is why it's very important to know where the product is marketed and how it is registered, because in general, LVPs are registered as drug product. However, in certain cases, they can be registered as hybrid or as devices, and therefore certain aspects for devices should be followed here in this bucket. So what can be critical is having certificates or compliance for food contact material per EU or 21 CFR, to have the compliance from pharmacopoeia standpoint, EP 3.1 or USP 661.1, the biological evaluation of the ISO chapter, 10993, and the specific chapters for it, if we have an additive, if we can have the 21 CFR 177 or adhesive for the 175. It's also important to get the values for EU 10 2011 and its amendments for the monomers and additives, chapter 3.2 for closures, 381 USP for the rubber. Also, we can, if there is a print or an ink or on the label, it will be important to get the specific requirements for that, or the heavy metals per EU, or ICH Q3D if is possible. And if we have PVC, it will be very helpful to get the compliance with EP 3.1.7. So this point, now in the project, if we start following it from exploratory phase, we are collecting all the information, all the supporting data that will allow us to perform a paper risk assessment. We know now what is the material of concern, what we can expect, and this will help us move to the second step, which is mitigating the risk. So to mitigate the risk, we eventually have to perform an E&L testing. And in this case, the following should be considered. First, select a test article. So what kind of test article we need is a worst case scenario where the bag exhibits the highest material to volume ratio and it is the lowest volume. Defining the AET based on the maximum daily dose and the treatment period for acceptable intake, selecting extraction solvents that are compatible, that are simulating the actual drug chemistry from aqueous, pH, and polarity, defining extraction time and accelerated storage based on clinical use and storage conditions, defining sterilization conditions, because it's important to consider the worst case scenario in this case for steam sterilization, for example, for 1 hour at 121 degrees, but also maybe the port tubes are gamma sterilized. Also, we have to consider each and every component and each aspect of the sterilization in the test article. And finally, we have to select appropriate analytical techniques that exhibit high sensitivity and accuracy to allow us achieve the detection limit based on the defined AET. So generally speaking, for extractable testing, it can be divided to organic and inorganic. For inorganic, ICP-MS analysis would be a great choice, while for volatiles, semi-volatiles, and non-volatiles, we can do testing by GC, headspace GC, GCMS, and LCMS using non-validated methods, just a screening against the AET value. And once we generate the results, all the results above this AET should be submitted to toxicological assessment. At this level of the project, it boils down to the toxicological assessment and what we define as margin of safety. So the margin of safety will put us in front of two scenarios. If the margin of safety is high, so we are expecting that the closure or the closure containing system of the LVP will not pose any risk to the patient, and we just need some confirmatory testing to demonstrate safety. We possibly can do a leachable study on the finished product, maybe on stability batch, for example, that is passing through the actual manufacturing situation cycle and at the end of shelf life to demonstrate safety. However, if we have low margin of safety, around 1, then a need of leachable study is required. And then first we have to start by doing a feasibility study to count for the matrix effect, have a validated method for the leachables using ICHQ2 guidance, and finally monitor targeted leachables during stability. What are the requirements that allow us to take an extractable to become a targeted leachable? It's highly recommended that the targeted leachable should first and foremost have a low margin of safety, represent different parts of the closure system, maybe parts one target leachable representing the primary film, another one the prints, another one the closures, some leachable that can represent a whole family. For example, if we can take Erganox 1010, can represent all the Erganox degradants as for the antioxidants to that matter. And finally, it has to be feasible to measure, and we have confidence in detecting it every time during the stability batches. So here, by now, we have all the information to submit the file, get the regulatory approval, and we have controls in place. Now, what will happen next? Product is launched, product is on the market. we have to make sure that we have a plan to cover the life cycle of LVP from E&L standpoint. What I'm presenting here is lifecycle management decision tree that we use internally and will allow us to handle the change controls of the products on the market based on a risk assessment approach. So when there is a change in an LVP, not necessarily every time we have to perform testing. We can classify the change into minor, moderate, and major. Minor changes, for example, a new label on the tertiary packaging, on the carton box of the product. There's no possibility, more or less, that there is a migration from the carton box to the solution product, and therefore the probability is very low for migration, and we can rank this change as minor, and no testing is required. Moderate changes are a little bit more tricky because they can pose a change, but then we have to evaluate this change to see if we need to do testing or we can support the safety by a solid scientific rationale. For example, if there was a pot tube change where the resin in the outer layer of the pot tube changed in the resin or a catalyst, but it's not in solution contact, and the supplier provided us with all the supporting data needed, and we have in-house a good knowledge about the full E&L profile for this product, it's possible to grade the severity and probability of the risk, a risk of a new leachable or a higher concentration of a leachable that may migrate to the solution. Based on this risk matrix, we will be able to evaluate if we need to perform testing or not. Third category is major, and this is pretty straightforward. If a change is classified as major, such as a change for the resin or adding an adhesive or changing to the adhesive in the primary film of the LVP, we will directly perform E&L testing as explained before. And sometimes these kind of testing can be triggered by regulatory gaps or new compliance, new guidance. For example, the nitrosamine guidance. And now I will explain how we have to deal with nitrosamine from LVP standpoint. What are nitrosamines? So nitrosamines can be formed when we have secondary, tertiary, quaternary amine, like amides, carbonates, amines that interact with nitrite or nitrosing agent, perhaps NOx in the air, in an acidic condition and form these nitrosamine molecules that can disrupt the DNA. In LVP, nitrosamines can come from three parts, if you will, of the product. So first, it can come from the solution, either the raw material of the APIs, the excipients, or during the manufacturing of that, from the closure system, and focus should be applied on the print labels and on the rubber, or during the manufacturing of those. And during the manufacturing process itself, maybe due to the presence of nitrite in the water quality in the plant, or using of solvents that has residual solvents in them during mixing or during sterilization, or the combination of these aspects together. So the guidance for EMA and USP was pretty clear, and the medicinal companies need to submit risk evaluation for their products on the market by end of this month, March 2021. And we have to submit an assessment that yes, we have a risk, no, we don't have a risk. A maybe statement or a low risk statement is not accepted by authorities. And if a risk is identified in this step, the medicinal companies needs to go to step 2. And in step 2, we have to perform confirmatory testing to understand where this nitrosamine is coming from and put controls in place and make sure that the products on the market are compliant by September next year. However, during that process, if any change takes place for, say, manufacturing process or raw material or packaging, we have to submit this information and these changes to the authorities. From LVP standpoint, let's say we detect a potential risk in an LVP product, and we need to understand from where this risk is coming from. So it's possible to start by performing screening study to identify the root cause, to understand from where this risk is coming, to allow us to put controls in place and mitigate this risk. So the nitrosamine detection limit per the guidance is required to be based on 30% of the analytical evaluation threshold. And how we have to calculate the analytical evaluation threshold based on the AI divided by the maximum daily dose registered on the SABC file. So the acceptable intake value can be derived from the list of the different nitrosamines on the guidance. This is the case if we expect one nitrosamine to be in the solution. In the case we have multiple nitrosamines in the solution, we have to select the AI of the most potent nitrosamine. In this case, for the defined one, is the 26.5. However, if we have other nitrosamine molecules, we have to go with a value of 18. Let's imagine hypothetical scenario for an LVP product. Say that we have an LVP with 2 liters MDD and we have multiple nitrosamines in this solution, or we are expecting to detect multiple nitrosamines in this solution. The limit, that reporting limit, goes down to 2.7 nanogram per liter. This is quite a challenging analytical limit for the sensitivity of the method. And this has always been a challenge for LVP, specifically for unknowns. But in this case, it's not an unknown. It is an identified molecule. It's a targeted molecule, and we can get around it using the appropriate techniques. So the LVP finished product can be extracted, concentrated to achieve this limit, spiked, and analyzed, and the regulation recommends to analyze LC-MS-MS. And we have an understanding if there is nitrosamine and from where it's coming. However, when we are in this step, we have to follow it up by confirmatory testing using a validated method and a follow-up step, using selective method that should demonstrate high accuracy for limit of quantification for each and every nitrosamine molecule on the finished product. And when we report the data, we have to report the data as a margin of safety concept by using the acceptable intake of the most potent one divided by the sum of all the measured nitrosamine in the solution. And for this, we'll follow up by a control strategy. Now we identify the risk, we know where it's coming, we know how it's behaving, we know what's its fate in the product. Then we can perform testing on 10% of the annual batches at regular basis. It seems simple, however, it's quite complex because the LVP have a lot of different drug chemistries and therefore a lot of different matrices. The complexity of the matrices made this step quite complicated. And this is a big challenge to achieve these limits with this extraction step for these matrices. And the other part, of course, is the unknown, and I'm sure this will be covered in another talks in this symposium. I will stop here, and just as a take-home message, I will really encourage that the ENL SME should be involved in the LVP design to ensure that there is a robust and successful qualification in place with a proper ENL risk assessment and adequate ENL study design that can qualify this LVP and manage it through the life cycle. And as a last call of action, from my personal experience, I noticed that an effective collaboration between medicinal companies and the raw material suppliers will greatly help and position the ENL strategy for LVP for success and allow us to tackle the challenges together.